Hello everyone, Mr. Linder here. Let's talk about digestion and absorption in the digestive system. So up to this point, we've been talking about secretions and how those secretions can help to facilitate the overall digestive process. The idea behind digestion is that we want to be able to break macromolecules into absorbable units that we can then bring into the cardiovascular system or into the lymphatic system. In order to accomplish digestion, we look at things such as mechanical digestion and chemical digestion. Mechanical digestion involves increasing the surface area of the molecules so that we can have better chemical enzymatic digestion take place within the lumen of the GI tract. In essence, we are working to break down polymers into monomers. And so mechanical digestion, such as chewing or churning in the stomach, is going to help to increase that overall surface area and make it easier for enzymes to get access to the macromolecules. The enzymes that are secreted into the GI system will work at varying pHs depending upon what location they're secreted into the GI lumen. For example, salivary amylase will work at approximately a pH of 7, so approximately 7 for salivary amylase, because this is released into the oral cavity, and the oral cavity has more of a neutral pH. So the optimum for salivary amylase is around a pH of 7. However, in the stomach lumen, we're looking at a pH optimum of around 2 for something like pepsin. Pepsin is a protein digesting enzyme, and it works well in an acidic environment because it's released into the stomach lumen, so it's part of the gastric juices. And then, of course, you have enzymes that work in the small intestine, like the duodenum. They're going to work predominantly around a pH of 9. So you're going to have enzymes like trypsin, which prefer that more alkaline pH. You'll have pancreatic lipase. And these enzymes that are secreted into the duodenum are going to work at that more alkaline pH because as the acid comes out of the stomach, it's being buffered by the bicarbonate that's dumped from the pancreas, and therefore you have more of an alkaline pH. So these enzymes need to work at those optimums. So depending upon where you are in the GI system, you're going to have varying pH optimums for enzymatic activity to be most effective. Most absorption takes place in the small intestine, and the reason the small intestine is a great place for absorption is because it has anatomical features that allow for an increased surface area. And the more surface area you have, the easier it is for nutrients, ions, water. It's easier for those things to get access to the epithelial cells and be absorbed through those epithelial cells into the cardiovascular or lymphatic system. So the anatomical features that we see in the small intestine are going to be things like villi and microvilli. So this diagram represents a villus structure. So this is the villus structure. And you can see that this villus structure is essentially an extension of the mucosa and submucosa. And this villus will have all of these epithelial cells that line this structure. And so if we were to enlarge the epithelial cells, we could see that the epithelial cells actually have microvilli. And so microvilli are actually extensions of the plasma membrane of the epithelial cells. And all those added extensions are creating more surface area for that epithelial cell. So this is increased surface area, right, to go along with the increased surface area that you see because of each villus structure. So if you have multiple villi, you have all these epithelial cells with microvilli, you are in essence enhancing the surface area along the small intestine. And so it makes the small intestine a great place to see most of the absorption that takes place in the digestive system. Notice also in this anatomical structure, you can see the lymphatic system. We can also see the cardiovascular system, arterioles, and of course venules. And so those are the structures that are going to receive the absorbed material, whether again it's ions or water or the various monomers, and then we're going to distribute those to the rest of the body. So again, the small intestine is a prime example because of its increased surface area to have better absorption in the GI system. Digestion and absorption is influenced by the motility in the GI tract. It's also influenced by all of the secretions in the GI tract, and it's going to be regulated by hormones, local control systems, and the autonomic nervous system. So what I want to do now is focus on the different types of digestion and absorption that we see. And so I want to start with the idea of carbohydrates being digested. So this is just a summary diagram. So this portion up here is related to carbohydrate digestion and absorption. And then this portion down here is for protein digestion and absorption. We're also going to look at lipid digestion and absorption, and then we'll look at some water absorption, salts, and some important ions and vitamins that get absorbed into the body as well. So let's start with the idea of carbohydrate digestion and absorption. So we're going to zoom in on this. So in carbohydrate digestion and absorption, we're trying to take the large polymers, so things like starches and glycogen, so large polymers, and we want to be able to digest those down into smaller structures. Ideally, you want to get all the way down into the monosaccharide structure. So we're going to take the polymers, the polysaccharides, and we're going to digest them down into disaccharides, and then we're going to eventually digest them down into monosaccharides, single individual monomers. About half of our calories that we ingest actually come from carbohydrates. So it's important that we're able to digest these polysaccharides into smaller structures and then be able to absorb them into the body. Polysaccharides and disaccharides need to be digested because in order to have intestinal absorption, it's limited to being able to absorb the monosaccharide structure. So you have to break down the larger structures or you're not going to be able to absorb any of those nutrients. We are actually unable to digest cellulose. You're probably familiar with that. We are unable to digest essentially this plant cell wall material, and that's because we are lacking the necessary enzymes to digest the cellulose. And so cellulose is actually what we would consider fiber in our diet. This is the dietary fiber that actually gives our fecal matter the bulk that it needs to have proper motility through the intestines. So without the proper enzymes, we're not able to digest this material. So if we're able to digest starch, glycogen, maltose, sucrose, lactose, that means that we actually have enzymes to be able to perform that digestion. And so we refer to that as chemical digestion. So we begin carbohydrate digestion in the oral cavity. In the oral cavity, we produce salivary amylase. Okay, so salivary amylase is produced in our mouth. It's part of our saliva that comes from our salivary glands. And it's going to start the process of breaking down starches and glycogen into smaller structures. So essentially breaking it down into disaccharides. Salivary amylase works at a pH of 7, so that's its optimum. once you swallow and move material down the esophagus and into the stomach, you're going to see a denaturization of salivary amylase because it's not going to perform well at a pH of 2. It's going to be too acidic for it. Carbohydrates then don't complete their digestion until you move into the small intestine. So there's very little digestion that can occur in the stomach, and so we have to move on to the duodenum. Carbohydrates reach the duodenum, and they are digested by pancreatic amylase. So you have a secretion from the pancreas of pancreatic amylase, and that amylase now can continue to digest starch and glycogen and break that down into disaccharides. And then we get further digestion of the disaccharides by what are called brush border enzymes. The brush border enzymes are enzymes that are attached to the epithelial cells, and they would literally be on the microvilli structures that we just talked about earlier. So if this is an epithelial cell, you're going to have enzymes that are embedded in the plasma membrane. So if we have the maltase enzyme, so we're going to have enzymes embedded in the plasma membrane of the epithelial cells, and those enzymes then are going to be able to break down further the disaccharides that have entered into the duodenum. So any enzyme that is attached to the microvilli is going to be referred to as a brush border enzyme. Okay, so the brush border enzymes. And it's not just carbohydrate ones. You're going to see other enzymes that are brush border enzymes. But we're starting with this idea of carbohydrate digestion and these carbohydrate brush border enzymes. So the three brush border enzymes that are of interest to us are maltase, sucrase, and lactase. And these three enzymes will break down the disaccharides maltose, sucrose, and lactose. So we'll take maltose and digest using maltase to form 2 glucoses. We'll take sucrose and digest using sucrase to form a glucose and a fructose. And then we'll take lactose and digest it with lactase to form a glucose and a galactose. And so now we have these monosaccharide structures that are going to be easily absorbed. So how do we absorb these monosaccharides? Once you're able to digest using amylase and using the brush border enzymes, how do we get absorption in the small intestine? So glucose and galactose are going to utilize a sodium glucose transporter. It is a symporter. So here we have sodium, and then here we have the glucose or the galactose, and we can see that they're both moving in the same direction. So this is a symport. And so we're able to absorb the monosaccharides of glucose and galactose as we transport sodium across the epithelial cells. Fructose will actually utilize a GLUT5 transporter. It's a glucose transporter on the apical surface. And then that fructose will move across the apical surface, and then it will diffuse through the epithelial cell. And then on the basal side, it will use a GLUT2 transporter. So this is our GLUT2 transporter seen here. This one up here is the GLUT5. And we're able to move that fructose from the luminal space across the epithelial cell and then, of course, into the extracellular fluid, or we could say the interstitial fluid. But ideally, we're getting the glucose, the galactose, and the fructose to move into the cardiovascular system. So we're actually moving these sugars into our blood capillaries. So the epithelial cells are interesting in that as they're moving the glucose across the cells, they don't actually use that glucose as a primary source of energy. And that's a good thing, because if we were to bring in the sugars into these epithelial cells and then they start taking that glucose or galactose and turning it into ATP, then we're not going to be able to move those sugars into the cardiovascular system. So because they're not the primary source of ATP production, we can get plenty of sugars into the cardiovascular system. Instead, these cells are going to use amino acids, specifically glutamine, as their primary source of energy. And that will conserve the sugars and allow those sugars to diffuse and move into the cardiovascular system. Let's move on to protein digestion. So in protein digestion, we're looking at generating the monomers that we call amino acids. So we're going to be using some other types of enzymes for this. This diagram actually is a brush border enzyme diagram. So we just finished talking about the brush border enzymes, sucrase, maltase, and lactase. But you should also, again, be aware that there are other brush border enzymes. For example, aminopeptidase, enterokinase, and these are going to be important for proper protein digestion. We have other enzymes. We have enzymes from the stomach. We have enzymes from the pancreas that are also used in protein digestion. But I wanted you to be aware that we are also using some of these brush border enzymes as well. Those enzymes that are attached to the plasma membrane of the epithelial cells. So here we have sort of the large overview diagram of protein digestion. In this diagram, you can see both the digestive process. So we have enzymes that are used for digestion, and we're going to talk about endopeptidases and exopeptidases. And then on this diagram over here, we can see the concept of absorption. How do we move the smaller molecules across the epithelial cells and into the blood, into the cardiovascular system? So I want to enlarge this and focus on one thing at a time. So first thing to really consider or talk about is that really not all proteins are equally digestible within the intestine. So some proteins will be digested easier than other proteins. For example, it's harder for the human digestive system to digest plant proteins compared to, say, animal proteins. And so there's going to be situations where some of the protein material is not completely digested. And sometimes we actually get endocytosis of larger peptide fragments into the cardiovascular system. And we'll come back to that concept in a little bit. So not all proteins are digested equally. But in order to digest proteins, we need both endopeptidases and exopeptidases. So what are we talking about here? Well, endopeptidases are going to be things like pepsin, trypsin, chymotrypsin. These are enzymes that are going to break larger proteins, so larger polypeptides. So this is a polypeptide structure. They're going to take the larger polypeptide structures and fragment them internally. So, chopping in the interior of the molecule. And so what that does is that is going to release, smaller fragments. So maybe you release a tripeptide or a quaternary peptide, depending upon where the fragmentation takes place. You might even get some dipeptides from that as well. So endopeptidases are working on the interior. So again, think of enzymes like pepsin from the stomach, trypsin from the pancreas, chymotrypsin also from the pancreas, elastase. These are all endopeptidase type enzymes. Then we also have the exopeptidase enzymes. Okay? These are vitally important to our ability to absorb amino acids into the cardiovascular system. And that is because the exopeptidases, and the two varieties are aminopeptidase, which is one of those brush border enzymes, and carboxypeptidase, which is a pancreatic enzyme. These are going to work at the ends of the peptide structure. So aminopeptidases work from the amine end. So you can see here we have the amine end of the molecule. So aminopeptidase is chopping from the amine end. And this allows us to generate single amino acids. And then the carboxypeptidase is working from the carboxyl end. This is the carboxyl end. So that C double bonded oxygen OH group. And so we're able to digest from the carboxyl end of the peptide structure. And again, that creates a single amino acid. So we get these single amino acids breaking off from the peptide structures. Once you have those single amino acids, that's going to make it a lot easier to absorb through the epithelial cells and into the cardiovascular system. So you have to have both endopeptidases and exopeptidases. Again, the endopeptidases chopping up large polypeptides into smaller fragments, and then the exopeptidases actually releasing the single amino acids that we're going to use for absorption. So let's go over again some of the enzymes that we see that work on proteins. So I mentioned the enzyme pepsin. The stomach is actually producing pepsinogen, and pepsinogen is being converted in the stomach into pepsinogen, into pepsin. So pepsinogen is the precursor molecule. It's what we refer to as a zymogen. So pepsinogen is activated by hydrochloric acid to produce pepsin, and this is taking place in the stomach. So because this is a stomach enzyme, it prefers that pH of about 2, and it works very well in the gastric juices. And it's an endopeptidase. It's working to take big polypeptides and create smaller fragments. Pepsin, though, becomes inactive once the gastric juices make their way into the small intestine. So once you're in the duodenum, you no longer have the activity of pepsin, and you have to turn to other enzymes. So those other enzymes are going to be pancreatic enzymes or brush border enzymes. So the pancreatic enzymes are going to start with the enzyme trypsinogen. So I'm going to go to a different diagram here. So this is showing you the various enzymes that are in pancreatic juice. So the protein digesting ones are going to be the trypsin, the chymotrypsin, the elastase, and the carboxypeptidase. So these are your protein digesting enzymes. The trypsinogen is also a zymogen. It's an inactive protein, just like pepsinogen. So pepsinogen had to be activated by hydrochloric acid. Trypsinogen is actually activated by a brush border enzyme called enterokinase. So enterokinase is in the plasma membrane of the epithelial cells in the duodenum, and that is activating trypsinogen into trypsin. Then you'll notice that the rest of the protein digesting enzymes, chymotrypsinogen, proelastase, and procarboxypeptidase, are all activated by trypsin. So trypsin becomes the activator. It's really important that we don't just produce active protein digesting enzymes because those protein digesting enzymes, if they were to get free and circulate through the body, would be going after all of the proteins that we use as structural components of the human body or as enzymes, hormones, neurotransmitters, etc. So we keep these protein digesting enzymes in an inactive form until they are dumped into the lumen of the GI tract, which is still the outside of the body. So we're doing this protein digestion in the external environment. So let's take a look at the activation of trypsinogen. So this diagram shows trypsinogen coming from the pancreas. It's an inactive zymogen. It's coming in contact with the brush border enzyme. This is the brush border enzyme that we call enterokinase. And so enterokinase is the activator. so that we can produce functional trypsin. And then trypsin acts as an endopeptidase to be able to break down polypeptides and create those smaller fragments that then can be further digested by aminopeptidase and carboxypeptidase. Once you have active trypsin, then the active trypsin can go on to activate chymotrypsinogen, procarboxypeptidase, and proelastase. And so we end up with the functional form of those enzymes, the trypsin, the chymotrypsin, the elastase, and these will continue to help us fragment polypeptides and, of course, create free amino acids using the carboxypeptidase and then, of course, the aminopeptidase. So carboxypeptidase and aminopeptidase are generating those free amino acids that we can then absorb across the epithelial cells. So lots of enzymes from the pancreas that we use to digest proteins. So again, trypsin, chymotrypsin, elastase, and then of course this really important one, carboxypeptidase, which is one of those exopeptidases to create those free amino acids for absorption. So how do we absorb the amino acids? So how are we absorbing these protein fragments? So most of the absorption is absorbing free single amino acids. So here we have amino acids, right? And we have this large arrow here to represent that most of the digestion is to create single amino acids, and then most of the absorption then is to absorb those single amino acids across the epithelial cells and into the cardiovascular system. So how do we accomplish that? Well, you can see right here we have a sodium and amino acid symport. Turns out, if you can absorb sodium is oftentimes the primary driver for moving things across epithelial cells. So if you can transport sodium, you usually can bring lots of other things with it. Whether it was sodium with glucose, now it's sodium with amino acids. And so we are bringing these across the epithelial cell on the apical surface. Once the amino acids diffuse across the epithelial cell, then you'll notice that we have an antiport on the basal side. Okay? And so the amino acid is going out as the sodium comes in. So we have a symporter on the apical side and we have an antiporter on the basal side, and then we move those amino acids into the cardiovascular system. Okay, so we're putting amino acids into circulation. There is some absorption. You'll see some of these smaller arrows here. So this arrow here and this arrow here. There is some absorption of dipeptides and tripeptides. The larger fragments, these di and tripeptides, they're going to be using a hydrogen dependent symport. So this is also a symport here. So as the hydrogen enters into the cell, it's bringing the dipeptide or tripeptide with it. We can digest some of those in the epithelial cells, creating single amino acids that can move into the cardiovascular system, or we can use an antiporter on the basal side. So here's antiport. We can use an antiporter with hydrogen to move some of those dipeptide fragments into circulation as well. There is a much smaller percentage of larger peptides that are absorbed into the cardiovascular system. And the reason being is that if we are to absorb larger peptides, there is a chance that these peptides can become antigenic and can actually trigger our immune system. But there is some transcytosis of larger peptides across the epithelial cells. So this would involve an endocytosis on one side, so endocytosis on the apical side of the cell, and then exocytosis on the basal side of the cell. So this is what we refer to as transcytosis, moving across the epithelial cell, requiring an endocytosis on one side and an exocytosis on the other side. Although this is a small amount of protein fragments, it does have this implication for medicine because some of these proteins can be antigenic. So intestinal absorption of large peptides may actually be a significant factor in why humans develop certain food allergies. So this could be why you may have a peanut allergy or a fruit allergy or even something like a gluten allergy from wheat proteins. So oftentimes we delay the feeding of infants allergy-inducing type foods just so that we can reduce the incidence of developing a food type allergy. So oftentimes pediatricians will recommend that you delay giving infants, peanuts or gluten containing wheat products just so that you can potentially avoid, or may avoid some of those allergies. This is not a guarantee. This is just, do this so that we can minimize the risk of developing those types of allergies. The next type of digestion and absorption to take a look at is the digestion and absorption of fats. So this particular diagram is a summary of what we're going to take a look at. So we are looking at the functionality of bile salts that are found in bile. We're looking at the micelle production and how that's involved in the digestion process. And then we're looking at the absorption of fats into circulation within the body. So about 90% of our fat calories come from the digestion of triglycerides. And triglycerides are found in both plants and animals. Other related lipids that you are digesting and absorbing would be things like cholesterol, phospholipids, and then, of course, things like fat-soluble vitamins, long-chain fatty acids, and so forth. But mostly, we are looking at triglyceride digestion and absorption. So that's mainly what we're going to focus on in this discussion. So how does this actually work? Well, the digestion and absorption of fat is kind of a complicated process because fats are not very water soluble. They're not water soluble. And so because fats are not water soluble, we have to find a way to make them more water soluble. And so in order to properly digest and absorb fats, we first have to emulsify the fats. So we're going to create an emulsion using bile salts. So bile salts are going to be our emulsifier. And a bile salt is really just a structure that has a hydrophobic side. So that's a water-fearing side, and then it has a hydrophilic side, the side that likes water. Your hydrophobic portion is going to orientate towards the lipid structure. So here we have the hydrophobic part orientating towards the lipid droplet. Your hydrophilic portion is going to orientate towards the water molecules. And so what this basically does is this allows us to take large fat droplets and create smaller fat droplets out of them. Okay? So the emulsification process actually increases surface area so that it's easier for chemical digestion. We're making it easier for chemical digestion. Making it easier for enzymes to get access to the lipid droplet. So what is the bile salt essentially doing? Well, it's orientating its hydrophobic and hydrophilic structures so that it can extract out these droplets. So for example, we're going to take out a droplet here and make a smaller one and make it more water soluble. So if you were to look at that droplet, you would have a bile salt structure, orientated with its hydrophobic portion towards the inside and its hydrophilic portion out towards the water side. And that's going to allow us to create these smaller droplets so that we can have better chemical digestion. So the chyme, which is the material from our stomach that enters the small intestine, is going to be emulsified within the small intestine because that's where your bile is coming from, right? Your bile is coming from the gallbladder where it's being stored. It's coming down the common bile duct and it's going to make its way into the small intestine. So bile is performing its emulsifying properties inside the duodenum. And the bile salts are able to do this because of that amphipathic nature that they have, meaning they are both hydrophobic and hydrophilic. And so what are we creating? Essentially what we're creating is we're creating a micelle structure. And so the micelle structure is basically how you're going to see the digestion of the fats by the lipases. So once you create those smaller lipid droplets, then you can have colipase and lipase come from the pancreas, being dumped into the small intestine, and it's going to aid in the digestion of the fats that have formed in this smaller lipid droplet, and you're going to create this micelle structure. And so this is what we're looking at here. This is a micelle structure where you have bile salts, you have phospholipids, you have fatty acids from the breakdown of your triglycerides, you've got cholesterol in here, and so forth and so forth. So you're beginning to see some of this digestion that has occurred in this micelle structure. And that is coming from the activity of enzymes. So we have lipase. This is pancreatic lipase. That's from the pancreas. We have colipase, also from the pancreas. And these enzymes, now because you've had the emulsification process, these enzymes are able to get access to the triglycerides, and we begin to see the breaking off of free fatty acids and forming monoglyceride or even diglyceride type structures, and they're found in this micelle structure. We also have other enzymes, because I did mention there are other types of lipids. So we have prophospholipase. Prophospholipase is activated by trypsin to form phospholipase, and this is the enzyme that is used to digest phospholipids. You have cholesterol esterase, which also comes from the pancreas to help free up cholesterol molecules. And so we're seeing further and further digestion taking place. So it's not just the triglyceride digestion, it's also the phospholipid and the cholesterol digestion as well. Once you have all of these structures, then you start getting into the idea of absorption taking place. So quick little summary. So we have the bile salts that have come down from the gallbladder and have dumped into the small intestine. We have our large fat droplets that have come from the food material that we have consumed, and it's gone through our stomach, and it's mixed in the chyme, and it's entering into the small intestine. And so now we have the emulsification process. So we take the big fat droplets and we make the smaller fat droplets. And then we start to see the enzymatic activity. So the enzymes have gotten involved. So we have our pancreatic lipase, our colipase, our phospholipase, our cholesterol esterase. All these enzymes are getting involved, and this is where we get these micelle structures. Once you have these micelle structures with this digested material, you're going to see the micelles begin to move closer to the epithelial cells. And once we get near the epithelial cells, then we can start seeing absorption taking place. So the cholesterol from the micelles, this is a cholesterol structure here, the cholesterol is being transported into the epithelial cells. And then we also have the absorption of monoglycerides and fatty acids. So those are coming out of the micelle structure, and then they are moving across the plasma membranes into the epithelial cells. Once the monoglycerides and fatty acids have entered into the epithelial cells, you see something interesting take place. You see them recombining together. So we actually reform triglycerides. We had to digest them in order for them, to be absorbed into the epithelial cell, but then they get reformed back together. So you actually end up with your triglyceride structure. So think of your glycerol with your 3 fatty acids. And then you have your cholesterol that's been absorbed. And then we have protein within the epithelial cells. So what we see happening is we see the cholesterol and the triglycerides and this protein in the epithelial cells being combined together to form a new structure. And the new structure that we're forming inside the epithelial cells is this chylomicron structure. So chylomicrons are created inside of the epithelial cells. And then what happens is they get processed in the Golgi apparatus. So chylomicrons are packaged into vesicles, and then we move those chylomicrons to the basal side of the epithelial cell. So we've absorbed from the apical side, and then we're moving this newly formed chylomicron structure to the basal side. Once the chylomicrons get to the basal side, we have exocytosis that takes place. And so exocytosis releases the chylomicrons, and now the chylomicrons can be moved into the lymphatic system. And so that's different than what we've seen with carbohydrates and proteins. Carbohydrates and proteins ended up in the cardiovascular system. So we saw glucose and we saw amino acids ending up in the cardiovascular system. But we're seeing chylomicrons end up in the lymphatic system. So the lacteal, which is the lymph vessel in the villus, becomes a really important structure so that we can get chylomicrons, get fat material into circulation. Once the chylomicrons enter the lacteal, they're going to be taken through the thoracic duct, which is a lymphatic structure, and then that's going to dump into your venous blood supply. So the thoracic duct is merging with your left subclavian vein, and so we are bringing fat material into circulation using the lymph and venous system. Once your chylomicrons are in venous circulation, we have a new enzyme in our blood, which is lipoprotein lipase. Lipoprotein lipase is what is going to start the digestion of chylomicrons within the cardiovascular system. And this is where we start to see formation of things like HDLs, LDLs, VLDLs, and so forth, which stand for high-density lipoproteins, low-density lipoproteins, very low-density lipoproteins. So this lipoprotein lipase is starting the process of creating our lipoprotein structures in the cardiovascular system. There is some short-chain fatty acids that can find their way into the cardiovascular system, but that is a very small amount. So there is a small amount of fatty acids that can get directly into circulation, but the vast majority of fats is the chylomicron structure. We also have nucleic acid digestion that takes place. So basically our DNA and our RNA are digested by enzymes. We use the enzymes deoxyribonuclease and ribonuclease, which come from the pancreas. So deoxyribonuclease breaks down DNA, ribonuclease breaks down RNA, and these will generate nucleotides, and those nucleotides then can be absorbed into the epithelial cells and into the cardiovascular system. So we use a form of active transport, essentially, to move nucleotides into circulation. We don't talk a lot about nucleic acid digestion and absorption. simply because it's not used for energy purposes, but obviously very important to have the building blocks for DNA and RNA in circulation and moving out to cells. The last thing for absorption to take a look at is absorbing vitamins and minerals. Actually, before I do that, just want to point out this is the ribonuclease and deoxyribonuclease. So this is for RNA digestion, and this one's for DNA digestion, and they're from the pancreas. So this is, again, all these enzymes listed here are pancreatic enzymes to help you review. So the last thing to take a look at is absorption of just a few other things. So fat-soluble vitamins, A, D, E, and K, those are easily moved into circulation because they're going to follow the same routes as your fatty acids and monoglycerides and cholesterol do. So they are absorbed through the intestine along with fats. They can become part of micelle structures, move towards the epithelial cells, and be absorbed. So moving across the plasma membrane for fat-soluble vitamins is fairly easy. Water-soluble vitamins, things like C vitamins, your B vitamins, these usually use some form of mediated transport. The big exclusion for the B vitamins is vitamin B12. Your vitamin B12... So vitamin B12. Vitamin B12 actually uses something called intrinsic factor. Intrinsic factor is going to, it's basically a protein that's going to complex with your vitamin B12, and it's going to aid the absorption of the B12 across the intestinal walls. Intrinsic factor comes from your parietal cells in your stomach, and then it makes a complex with your B12 vitamin, and then it's later absorbed in the intestines. If you are not producing enough intrinsic factor, then you can have a vitamin B12 deficiency, which is known as pernicious anemia. And so we require intrinsic factor to aid in the absorption of B12. Iron and calcium. So iron and calcium are actually regulated by hormones to facilitate their intestinal absorption. In this particular diagram, iron is ingested in the form of heme. So we actually consume heme from meat products, or we get iron out of plant material as elemental iron. So from your animal products, basically anything with blood, you're going to see the heme that contains iron in it. And then if it's plant material, then we're going to see elemental iron. Heme actually gets transported across the apical side of the cell and moves into the epithelial cell. Once heme is in the epithelial cell, we remove the iron from it, and then the iron can be moved into the extracellular fluid. The iron that comes from plant material uses a symport. So we have a symport with hydrogen ions. So this is a hydrogen iron symport, and that's moving the iron into the epithelial cell. And then again, we're eventually going to transport it into the extracellular fluid. The way that iron is transported is using a transporter called ferroportin. So the ferroportin is the transporter used to move iron into our extracellular fluid. We regulate iron absorption using a hormone called hepcidin. So hepcidin is a hormone that's actually used to regulate the amount of iron that gets absorbed through the GI system. Hepcidin comes from the liver, and when iron levels are high, the hormone hepcidin is released by the liver. It's done basically when iron levels are high, and it triggers the epithelial cells to destroy the ferroportin transporter. So if we have too much iron, we're going to release hepcidin. It's going to block this transporter, and then obviously that's going to stop the movement of iron until we bring our iron levels back down. So the liver is monitoring how much iron we have, and that will control how much iron we absorb from the GI tract. Calcium absorption occurs either paracellularly, that means between the epithelial cells, or calcium can move across the epithelial cells. Calcium enters the epithelial cells on the apical side using a transporter. So we have, or I should say, using a calcium channel, an ion channel, and then it enters the extracellular fluid using either a calcium pump. So we have a calcium pump that requires ATP, or we use a sodium-calcium antiport. So we have absorption of calcium from the lumen, and then we have pumping the calcium or using an antiport to get it into the extracellular fluid. Calcium movement across the epithelial cells is regulated by the hormone calcitriol. Another name for calcitriol is vitamin D3. So this is essentially a vitamin D hormone. So calcitriol or vitamin D3 is going to affect the absorption of calcium across the epithelial cells. And so what calcitriol does is it increases the production of a calcium binding protein within the epithelial cells. And this binding protein is going to increase the amount of available calcium that is able to be actively transported or to be pumped, right? So this is active transport. It's making the available calcium available for active transport into the extracellular fluid. So we use calcitriol to essentially enhance intestinal absorption of calcium. So whereas hepcidin, right, prevented iron from being absorbed, calcitriol increases the ability for calcium to be absorbed. And then the last thing to consider is the movement of ions and water across the epithelial cells. And so most water absorption is going to take place in the intestine, the small intestine in particular, some of it in the large intestine. Water is going to follow the osmotic gradients. So if you can absorb these ions, if we can get ion movement into the extracellular space, okay, then we can have water following. Okay? So water is going to follow just based on osmotic gradients. And so in this diagram, you can see that there's quite a few transporters for things such as sodium. There's chloride transporters. There's symports using, there's symports using sodium and organic molecules like glucose, amino acids, and water is going to follow those. There's antiports to bring sodium into the cell. There's antiports to bring chlorine into the cell. And so we have all these different transporters that allow us to move various ions across the epithelial cells or move organic molecules across the epithelial cells. And then, of course, the water can follow. So that's a little bit on digestion and absorption of various molecules in the GI system. I hope that helps. Take care.